September 19th, 8 speakers, 3 Nobel laureates, 3 to-be Nobel laureates. Is it? Difficult word, I guess. And 2 other people. So I will not tell you who are those other 3. And so today also is Friday, so we're celebrating vision research, but today will be with a little spin. It's not going to be that much about vision, but we wanted that technology and science to bring into our beloved retina and RPE and so on. All right, so with that, we have a speaker in-house, which is, again, wonderful, another thing. And with that, I would like to ask Meng to introduce our speaker.
Good morning, everyone. I'm Meng Wang, a research specialist from Dr. Richen's lab. It is my honor to introduce today's distinguished speaker, Dr. Samuel Sternberg, a leading scientist and innovator in the field of molecular biology. Dr. Sternberg is an esteemed researcher known for his groundbreaking contributions to the study of CRISPR gene editing technology. Currently, Dr. Sternberg is an associate professor in the Department of Biochemistry and Molecular Biophysics at Columbia University, where he leads his own cutting-edge research laboratory. His work focuses on CRISPR-Cas systems, RNA-guided enzymes, and genome engineering technologies, advancing our understanding of these transformative tools in molecular biology. His research has been widely published in top scientific journals like Nature, Science, and Cell, and has been covered in the New York Times, Science News, The Scientist, and various other news outlets. And his work has received numerous accolades for its impact in the field. Dr. Sternberg's academic journey began at Columbia University, where he graduated summa cum laude with a Bachelor of Arts degree in biochemistry in 2007. He then pursued his PhD in chemistry at the University of California, Berkeley, working under the mentorship of Nobel laureate Dr. Jennifer Doudna, a pioneer in CRISPR research. And his contributions to the field have been widely recognized, earning him prestigious honors such as the NIH Director's New Innovator Award and the NSF Korea Award. He is also an investigator of the Howard Hughes Medical Institute. Beyond his groundbreaking research, Dr. Sternberg is deeply engaged in public outreach and ethical discussion surrounding genome editing. He has shared his expertise on plenty of international platforms. And in addition to scientific contributions, Dr. Sternberg is a passionate educator and communicator, co-authoring the book with Dr. Doudna titled A Crack in Creation, Gene Editing and the Unthinkable Power to Control Evolution, which provides a compelling account of development of CRISPR and its ethical implications. And the book has been described by the New York Review of Books as a required reading for every concerned citizen. His ability to bridge complex scientific concepts with real-world applications has made him a respected voice in both academic and public discussion in gene editing. Today, Dr. Sternberg will present in molecular innovation at the CRISPR-transposon interface, where we have the privilege of hearing from him as he shares his expertise and insights with us. Please join me warmly welcoming Dr. Samuel Sternberg.
Great. Thank you for that very wonderful introduction. It's a real pleasure to be here. I think I've never given a seminar before 9 a.m. at a university before. Also, my lab usually wakes up around 9:30 or 10, so it's really cool to see such a vibrant audience this early in the morning. So I've also never presented to a department of ophthalmology before. This is not going to involve any research relating to the eye, but sorry?
I'm sorry you didn't
Yes.
present it yet.
But I'm really excited to share some of the work we've been doing in the lab, focusing on a new area of CRISPR involving intersections with transposable elements, and then some recent work that's going to be a bit more basic, curiosity-driven, but I think opens up a new potential place to look for molecular engineering tools. So I don't need to tell this audience how CRISPR has really revolutionized precision genome engineering, not just with Cas9 nucleases, but a whole plethora of tools that have enabled everything from genome to epigenome editing and a number of other ways of manipulating the cell's genetic information. And I think, we're all inspired by this first commercially approved drug that is a functional cure for sickle cell disease. And I'm really looking forward to hearing about some of the clinical work that's being done here at UC Irvine. What got me excited about CRISPR as a PhD student, though, was its role as an adaptive immune system in bacteria. And so CRISPR, of course, evolved first in nature. It was something that humans borrowed from a natural family of immune systems that protect cells from mobile genetic elements, principally viruses that infect bacteria. And I'll just emphasize here that although Cas9 has gained the most renown for its application as a genome editing tool, bacteria, if you look across the phylogenetic diversity, they encode dozens of different CRISPR-Cas subtypes, many of which are now being deployed as either more compact editing enzymes or tools for manipulating RNA if we look at the RNA targeting CRISPR systems. But my lab started working on CRISPR, but then we quickly found ourselves moving even earlier in evolutionary time towards the place where CRISPR actually first evolved, which is the world of transposons or jumping genes. And so I've quickly fallen in love with the world of transposons, which were famously first discovered by Barbara McClintock in the early 20th century. And we now know play fundamental roles in shaping genome evolution, speciation, and a number of other major phenotypes like the coloration of flower petals or moth wing patterns, and even phenotypes like whether primates have or don't have tails can be ascribed to retrotransposon insertion events. So just to pivot our focus away from CRISPR to transposons, we need to talk about what these selfish genetic elements are in their simplest sense. And so I'm going to focus today first on DNA transposons, which are bounded by inverted repeat ends that I'll show with these maroon triangles, and at least one enzyme, a protein coding gene called a transposase, that's responsible for recognizing these transposon ends and then mobilizing this piece of DNA from one donor site in the genome to another target site. And so in this simple cartoon representation of a cut and paste transposition event, the transposase proteins recognize those ends, forming a synaptic complex that leads to DNA excision, and then another integration reaction where that mobile DNA is pasted elsewhere in the genome. A reason that transposons have really captured our attention in the lab is that if you look at the genes that are responsible for their mobilization, these are by far the most abundant genes in all of nature. As you can see in this profile of a number of different gene families across hundreds of analyzed genomes, transposases are just all off the chart as an outlier up here in the top right. There's also many different mechanisms to move DNA around the genome. It can be mobilized through DNA or RNA using distinct kinds of enzymatic domains. I'll focus quite a bit in the first part of my talk on whether they move with either specificity or in a random nature, and the extent to which they can associate with new genes towards their spread mechanism. And then to bring this back to humans, I always like to emphasize and remind folks that, of course, almost half of our own genome comprises transposons, principally retrotransposons that have mobilized using reverse transcriptase enzymes. So these are really major and powerful shapers of the genome. Now, transposons we think of as being very selfish in their desire to mobilize and proliferate in the genome. But beyond host organisms evolving ways of silencing or repressing transposons, I want to shift your focus to appreciate a different kind of equilibrium between transposons and host organisms, where there's actually a rich mutualism between these two, where the genes from transposons can serve as a rich reservoir for the host to evolve new functions by co-opting those genes for new biological pathways. And so we think about this abundance of transposons as genomes and the diverse ways they move as serving as the starting material for the cell to co-opt those same enzymes for pathways as varied as building immunoglobulin genes through VDJ recombination, developing the ability to splice messenger RNAs or maintain telomeres. All of these use enzymes and enzyme parts that came from simple selfish genetic elements. And so to hit that point home, I want to define a term that was first coined by Stephen Jay Gould and Elizabeth Verba back in the 80s, exaptation, which is a bit different than adaptation in that it refers to a gene shifting its function during the course of evolution. And we see this happening over and over again from genes stolen from transposons being repurposed for new functions. So I already gave 3 examples. Transposases from DNA transposons being repurposed to promote the DNA cutting events that install recombined immunoglobulin genes, the self-splicing ribozymes in group 2 introns being repurposed for the spliceosome in eukaryotes, and reverse transcriptases and retrotransposons being repurposed to promote telomere synthesis in eukaryotic chromosomes. And so inspired by these examples and some of the work in the lab, we really have sought to discover new mechanisms where molecular functions have evolved at this critical interface between the host and between transposons. And what I'm going to tell you about today in 2 stories are 2 kinds of RNA-guided tools or enzymes that have emerged at this interface between the host and transposon. So the first vignette is more or less what the title of my talk was about, a new link between CRISPR proteins and transposases that promote a new type of chemistry, RNA-guided DNA integration. And then in the second story, I'm going to tell you about something we recently published in which reverse transcriptases that came from retrotransposons have been co-opted as a new way of templating the synthesis of genes in the cell towards an antiviral immune response. And I'd be very happy, by the way, to be interrupted if there's any questions that come up during the talk, so feel free to raise your hand if you'd like. Okay, so this first story began when we started looking at a family of transposons in bacteria within the TN7 superfamily that had a very peculiar feature, which is that in addition to transposase genes, an analysis of the other genes encoded within the mobile DNA uncovered the presence of CRISPR-Ras and CRISPR-associated genes. But there was one very peculiar feature of these systems, which is that the Cas genes never had the intact nuclease domain functionality. And so what we were left with was an array that could generate guide RNAs and Cas proteins that could, in theory, use those guide RNAs to target DNA, but not cut. And so we hypothesized that this might represent an intact DNA targeting module which could work together with the transposase DNA integration module to promote this new kind of chemistry, RNA-guided DNA integration. And so we set out to test this hypothesis, focusing on this new family of transposases that I'll be calling CRISPR-associated transposases, or CASTs, for the rest of my talk. So this is going a number of years back now, and work led by a fantastic former PhD student, Sanne Klumpe. She focused on a candidate CAST system that we sourced from Vibrio cholera. I'm showing you here the genetic architecture at the top. At the very left and right ends are the transposon boundaries. There's a number of other cargo genes in gray that we skipped or ignored for the moment, and instead focused on the genes in blue, the CRISPR genes, the CRISPR array, and then the transposase genes. And Sanne hypothesized that if we could develop a cellular system for reconstituting this machinery, we might be able to study this hypothetical process of RNA-guided transposition. So she developed an assay to do this in E. coli, where we had a piece of donor DNA on a plasmid, guide RNAs targeting sites in the E. coli genome marked here 1 or 2. And then we initially used PCR to detect the presence of these potential integration products by looking for unique amplicons formed only in the presence of matching guide RNAs. And some of her first results, the first results of the lab really, she observed a very robust and clean amplicon only with a guide RNA or CRISPR RNA targeting site 1 and the presence of all 3 other plasmids. And that band was missing in any of these negative control conditions. But she then subjected that amplicon to Sanger sequencing. We found something very interesting, which was that unlike Cas9, which cleaves and edits DNA within the region targeted by the guide, here the guide RNA was actually directing the payload to be integrated at a fixed distance downstream of the target. Exactly 49 base pairs from where the guide RNA matched the DNA was where the transposon was inserted, and it was inserted with characteristic target site duplications, a feature that is a hallmark feature of transposition. Now, after a number of other experiments, we of course wanted to ask, how does this look genome-wide? This was many years after a number of off-target profiling methods had been developed for Cas9. And so we developed an assay for profiling genome-wide DNA integration. We first tested it on a well-studied transposon used for random mutagenesis in bacteria called Mariner. Here we're sequencing in a large population of cells, all the DNA integration events and mapping those insertion sites back to the genome. So this is the E. coli genome, it's 4.6 megabase pairs. And you can see that Mariner, like most transposons, has no sequence specificity. And so the integration sites across the population are indiscriminately spread across the entire genome. The Cas system behaved fundamentally differently. So here with the guide RNA targeting the site marked with the triangle, we saw really only reads mapping to this target site and no non-specific transposition events anywhere else. So this was very exciting for Sana, and so she immediately said, can we reprogram the system just like you would reprogram a Cas9 nuclease? And indeed, when she tested a number of arbitrarily chosen guides, shown here on the right, to target other sites in the E. coli genome, in each experiment shown by each bar graph, the insertion events all beautifully moved to the new site in the genome matching the guide. And so together with a number of other experiments, this led to an initial model for how RNA-guided DNA integration operates in Cas systems. Target sites are recognized by a CRISPR complex that is more or less analogous to a nuclease-dead Cas9. It has a guide RNA, it requires a PAM, it requires RNA-DNA complementarity, but there's no cutting event. And instead, we identified and discovered a new protein component, TNIQ, which is the glue that connects the CRISPR complex to the transposase. And so the transposase then drags along the donor DNA, shown here in blue, and leads to the downstream integration of the donor in a double-strand break-free reaction. And this characteristic 49 or 50 base pair spacing we envisioned would be a function of the molecular footprint of these different protein components connecting the CRISPR module to the transposase module. And I'll just highlight here that over the years, we've tackled this from a number of different approaches, including structural biology. And so working with Israel Fernandez, Tyler was able to determine the cryo-EM structure of the first step in this pathway, where we could now see these new interactions between the CRISPR complex and the guide, shown in red, and this transposase component, TNIQ, leading to this eventual DNA integration reaction. So Cas, we think, represents an exciting new family of programmable recombinases that differ fundamentally from random transposases like Mariner or Piggyback, which are great for making random untargeted insertions, but can't be controlled in their specificity. And they also differ from site-specific integrases like BXB1 or FASI31, which are great at inserting payloads at landing pads, but can't be easily re-engineered to insert at new user-defined sites. Cas, on the other hand, are by their very nature programmable and RNA-guided, which we think gives them the kind of flexibility that we'd like to harness to build tools. So over the years, we've studied the basic structure, function, diversity of Cas systems, but at the same time, we've really wanted to apply them as a new type of genome engineering platform. And so in work that I won't talk about, we've been developing them in bacteria where we can actually now do very precise manipulation of species within a complex microbiome like the murine gut. But what I want to share with you is our progress to apply these in mammalian cells and human cells as a new approach to performing targeted gene insertion. And just to give a little bit of context on the reasons behind this goal, double-strand breaks are now quite easy to introduce using Cas9 nucleases, but editing with double-strand breaks still suffers a number of drawbacks. First of all, it's very difficult to control the otherwise heterogeneous products that arise from a competition between end-joining repair and homology-directed repair. And then in experiments where a researcher might want to install precise insertions or modifications, performing HDR is restricted to mitotic cells where the DNA repair factors are expressed. The efficiency of HDR generally decreases quite significantly with increasing sizes of the genetic payload. And then a number of studies have shown that double-strand breaks can cause more deleterious aberrant outcomes like translocations or large on-target deletions. And so the field, I think, is beginning to move away a little bit from nucleases to enzymes like base editors that Audrey's lab, of course, is pioneering, and prime editors, both of which are excellent at making small-scale changes in the single to tens of nucleotide range. And where we'd like to really bring casts into the picture are accessing this much larger editing space where we can make insertions on the kilobase scale. And one of our motivations here is that while base editors, prime editors, HDR is great for targeting genetic diseases that are largely caused by a single pathogenic mutation like sickle cell, the reality is that most genetic diseases arise from hundreds of different mutations, as shown here by this histogram, where you can see that among 500 or so genetic diseases, the vast majority have hundreds of mutations across all patients in the population. And so in this scenario, it becomes quite impractical to scale base or prime editors to hit each one of these different mutations, which are often only experienced by a very rare cohort of patients. And so we envision using a more universal strategy to integrate healthy cDNAs at the on-target site using a double-strand break-free editing platform like CAST. So can we do it? This has been one of the major goals of the lab over the last 4 years or so. We think casts have many of the desirable properties for this kind of application, but those properties come about because they rely on distinct components, a CRISPR module and a transposase module. Working together, they generate this highly accurate integration, but that also means we have the challenge of how to deliver multiple components to the target cells. And so George and Rebecca were two courageous PhD students that tackled this goal of reconstituting casts in human cells. I'm going to skip most of what they had to struggle through in the early phases of this project and just jump right to one of the punchlines, which is that we found that although this Vibrio cholera cast system that I showed you data for worked great in E. coli, it didn't work great in human cells. And in fact, we had to screen dozens of different transposons with this TN ID number to find one very far away in phylogenetic space from a pseudo-Alteromonas species that had the starting activity in human cells that we thought was a better place to engineer from. And using this PSC cast system in a study that we published a year and a half ago, we were able to finally achieve in the tenths to low single-digit percentages of editing with this eCast3 version across about a dozen different genomic target sites. So it was a promising start, but we had a long way to go. And so we thought, how can we further improve cast systems for human cell applications? These are not separate from each other and not mutually exclusive. But the one I'm going to focus on for the next couple of minutes was inspired by the hypothesis that unlike a Cas9 antiviral immune system, which nature has sculpted to be an extremely efficient DNA cutting enzyme because it's a life or death scenario for the cell, transposons don't have a life or death scenario to jump as fast as possible. In fact, they probably have been optimized to be suboptimal because they need to jump only enough to survive, but not cause a fitness cost on the host. And so we hypothesized that DNA integration in Cas systems might be limited by an inefficient DNA targeting or catalysis step that we could maybe improve using directed evolution in the laboratory, where we could basically create our own evolution scheme to hypermutate and select winning variants in a pool. And so for this, we were very excited to team up with David Liu and 3 fantastic PhD students in this group, Isaac, Simon, and Shannon. And this was also spearheaded by George in the lab. I won't go into all the details here, but we relied on a directed evolution scheme that David's group has developed called phage-assisted continuous evolution, or PACE. And the punchline is we use viruses and E. coli to create a selection scheme where only when DNA integration is successful do the viruses propagate, leading to further rounds of mutation and selection. And the way we built this circuit was to require integration of a DNA payload, shown here between the right and left end of the transposon, that would insert a promoter driving an essential viral gene called G3. So only if the DNA gets integrated is gene 3 turned on, and that only happens if the evolving Cas component has been improved during the directed evolution process. And I'll just mention here that PACE is valuable because it really accelerates protein evolution hundreds of fold compared to conventional approaches where you would do each cycle manually in the lab. So this was a major undertaking, and a lot of this credit goes to of course, the team of researchers in David's lab that did all the engineering on the PACE side. They stepped through many different campaigns of non-continuous or continuous evolution, and by the end had screened about 1,000 generations of this directed evolution process and selected about 10 mutations in the TNSB transposase that when we mapped it onto predicted structures lay at many of the critical interfaces between proteins in the transposition complex, TNS A or B, between monomers of that transposase, or at the predicted interface between the transposase and the DNA itself. But did these actually improve activity in human cells? All that evolution happened in E. coli. We needed to move those variants over to human cells to see if they were actually selecting for enhanced activity. And that's what I'm showing here on the bar graph, where we started with a wild-type Cas system that has not been engineered. And you can see in these experimental conditions, it's barely detectable above the negative control that has no transposase in the cell. And now, over evolutionary time, from the first campaigns to the last campaign, you can see this beautiful progressive increase in the efficiency of DNA integration at the HEC3 site, and here at 2 other sites, which showed similar levels of improvement over this evolutionary time scale. And collectively, across a number of sites, we saw about a 230-fold increase compared to the wild-type Cas system. So just to give you a kind of quick profile of some other applications where we've now used this evolved or evo-Cas. We can now integrate payloads at a number of different safe harbor sites like AVS1 or the ROSA 26 locus. We've been able to install payloads at the TRAC locus for potential use in CAR-T engineering. And then importantly, we can integrate payloads at a number of different genes that are implicated in loss-of-function genetic disorders that I mentioned earlier, like cystic fibrosis or Stargardt, where hundreds of different mutations exist across the patient population. And in some cases, these efficiencies are in the right ballpark to begin thinking about therapeutic application in a more clinical context. And then importantly, because these transposases have evolved in nature to mobilize very large payloads, we wanted to make sure that we weren't selecting against the ability to mobilize similarly large payloads in human cells. And so here, focusing on that ROSA 26 locus, we showed that evo-Cas could generate 10 to 20% integration efficiencies of payloads spanning hundreds to 10,000 or 15,000 base pairs in size. And again, compared to the wild type, which was barely detectable above a couple tenths of a percent. So I'm going to stop this section here and just summarize. We think casts really offer promise because they inherently rely on nuclease-dead CRISPR systems to integrate or paste DNA rather than using double-strand breaks. They're the first category of enzymes that naturally perform an RNA-guided gene insertion reaction. They do work in human cells with the same genetic and molecular requirements as bacteria. And we think that directed evolution, although EvoCast is not the end of the story, it's offered a very flexible and powerful platform to improve the activity. And we're now undertaking a number of further projects to continue moving this forward. And again, our eventual goal is, of course, a research tool to make large gene insertions for interesting biological projects, but then also potentially for these kinds of gene therapy applications involving target insertion. Okay, in the last 20 minutes, I'm going to switch gears almost completely. We're going to stay on the theme of repurposing genes from transposons, in this case, retrotransposons. And we're going to stay within the theme of CRISPR-adjacent systems that are antiviral, but switch over to kind of a very interesting new area of science that we've been excited about in the lab. And to motivate this, I'm going to come back to a picture that I usually share is the first slide of my talks, which kind of captures, again, what got me excited about CRISPR in Jennifer's lab when I joined in 2010, which was not gene editing, but the mechanism of antiviral defense that CRISPR systems provide to the millions of different bacterial species on Earth. But what I want to emphasize here is that although a decade ago, if you were to list the known immune systems found in bacteria, the list might not go more than 4, 5, or 6 lines deep. This has really exploded in the last 5 years, thanks to pioneering work from Rotem Sorek, Feng Zhang, and many others in the field. And so we think that there's now such a huge diversity of newly discovered antiviral immune systems that it's a really exciting place to look for new biology and new biotechnology. So again, a decade ago, this is taken straight out of a review on antiviral immunity, and you see 5 different categories. And this is now from a review 10 years later. I think it's just one of the figures that had all these different buckets for the newly discovered pathways of immunity. And I'm going to highlight 2 things here, which excite us. One is that many of these newly discovered immune systems in bacteria have surprising evolutionary ties to human immunity. And so this is revealing new connections between primitive immune systems across the domains of life. And then, as you know, restriction enzymes came from antiviral immune systems known as restriction modification. CRISPR enzymes came from CRISPR-Cas immune systems. And so we think it's almost certain that new biotechnologies are going to similarly flow from this treasure trove of molecular pathways. And so as we were kind of thinking about how to tap into this new field, we realized, the majority of immune systems have a common feature, which is that they encode nucleases to degrade the DNA of the virus. That's everything colored here in green. And of course, restriction modification, where restriction enzymes come from, these are by far the most common. About 90% of bacteria have these immune systems, 40% have CRISPR. But we were interested in all the systems that are in gray because they have unknown function. And we became especially interested in these systems known as defense-associated reverse transcriptases because they were kind of the counterpart to a DNA nuclease. They don't cleave DNA. They would, in theory, synthesize DNA. And we were really intrigued what would be immune protective about creating DNA in a cell during a viral infection. So different kind of mobilizing enzyme. These are not DNA transposases. They're reverse transcriptases that also evolved to mobilize jumping genes. And they do that by using an RNA template to synthesize DNA through reverse transcription. So retroviruses rely on reverse transcriptases for cDNA synthesis before the DNA gets integrated. I mentioned earlier that telomerase is a reverse transcriptase co-opted from retrotransposons that uses the tRNA for synthesis of telomeres. And then that 40-ish% of the human genome that comes from transposons were amplified using reverse transcriptase. The story's not so dissimilar in bacteria, although I think bacterial reverse transcriptases have received far less attention. But it's ancestral group 2 introns that are probably the progenitor of all of Earth's reverse transcriptases. These are bacterial retrotransposons that have been domesticated or co-opted or exapted, if you remember that word I taught you, many, many different times to form large families of non-mobile reverse transcriptases. And one of these families is implicated in phage defense and really caught our attention. And then just to kind of connect this back to technology, if you've used or read about prime editing, these rely on Cas9 nicases fused to reverse transcriptase. If you've used lentivirus to make gene insertions, those of course also rely on reverse transcriptase. And there's even newer tools that are coming onto the scene relying on the reverse transcriptase from non-LTR retrotransposons. So we thought perhaps looking at these bacterial reverse transcriptases might also have crossover potential for technology. But we started with this question of coming back to a viral infection. Restriction modification systems cut the DNA of a virus. CRISPR systems cut the DNA of a virus. What about RNA-guided or RNA-templated DNA synthesis would similarly protect against the virus? And so that was really the question that Stephen Tang, a fantastic MD PhD student, set out to test. How do these defense-associated reverse transcriptases, or DRTs, provide immunity? And then over time, we'll start pivoting to how we can exploit these new enzymes for potential new tech. So I'm going to tell you about one specific reverse transcriptase system known as DRT2. It comes from Klebsiella pneumoniae. And it's caught our attention because it's extremely simple in its genetic architecture. A single protein coding gene whose entire length is mostly comprised of the reverse transcriptase domain and an upstream non-coding RNA. So we wanted to ask the question, does this reverse transcriptase bind that RNA, and what DNA synthesis products does it produce in the cell? And so Stephen's idea was to use kind of a high throughput sequencing approach to answer both these questions by introducing a flag tag onto the protein, IPing the protein in cells with or without phage infection, virus infection, and then isolate either RNA or DNA and perform high throughput sequencing to profile all of its RNA ligands and its putative cDNA synthesis products. And we thought that this latter tool, cDNA immunoprecipitation and sequencing, or CDIP-seq, would be a really powerful way to profile the so-called reverse transcriptome of the cell. So the RIP-seq, looking at the RNA, revealed that the most abundant transcript bound by the reverse transcriptase was that non-coding RNA encoded right upstream of the gene. So that was maybe not all that surprising, but we were very excited to see that if you looked at which transcripts were being reverse transcribed, clearly that non-coding RNA is the winner. And so we wanted to look deeper at what about this non-coding RNA is leading to DNA synthesis, and what does that tell us about the mechanism of immunity? So I'm going to expand this cartoon on the left in the next couple of slides. So here again is a reverse transcriptase protein coding gene that's transcribed and translated into the RT. Here's our non-coding RNA shown in magenta. And we're going to first update the squiggly line to something that better reflects the extremely structured and conserved nature of the non-coding RNA, which evolutionary information told us has a number of highly conserved stem loops, as shown by this covariance model, suggesting that there are critical structural features that are probably being recognized by the reverse transcriptase. And so here I'm now updating that cartoon on the left. Next question is, which part of the RNA is being reverse transcribed? So we went back to our CDIP-seq data, which I showed earlier, but now we're actually looking at the coverage of reads along the reference genome encoding the reverse transcriptase and the non-coding RNA. So the non-coding RNA starts and ends here and here. And quite surprisingly, the cDNA had a very well-defined starting and ending point that didn't overlap with the ends of the non-coding RNA, suggesting that only a certain defined portion of the non-coding RNA is being used as a template by the enzyme. And so we updated our kind of verbiage in the lab to break the non-coding RNA down into the scaffold portion, which is serving probably as a platform to bind the reverse transcriptase, and then the template portion shown in light pink, where the reverse transcriptase is actually performing reverse transcription. So we wanted to ask more about how the cDNA product would provide immunity. And Stephen made the first of three, I'd say, critical insights that led to figuring out how this whole mechanism works. And that was by looking much more closely at the cDIP-seq data and the raw data, which revealed an unexpected feature. So most plots like this would suggest that the reads leading to that visualization would start and end at the boundaries of where the coverage begins and ends. But in fact, Stephen found that the raw reads from a sequencing data most often had extensions on either the 5' or 3' end that were not complementary to the reference genome. And so the mapping algorithm was clipping them and basically removing them from the alignment, making it look like the reads started and ended here, but in fact, they were always extending beyond those boundaries. And so Stephen asked, what is being synthesized if it's not matching the reference in the non-coding RNA? And so he looked at the sequence on the 3' end of these cDNAs and found that, really surprisingly, it was identical to the sequence at the 5' end of the cDNA. In other words, this told us that the products it's synthesizing look something more like this. We have two perfect tandem copies of the same cDNA that are likely stitched together head to tail. And if you took the same data and now mapped it to this, now there was clear alignment across that cDNA-cDNA junction that's not at all present in the non-coding RNA, suggesting that the reverse transcriptase is making tandem concatenamers of the same sequence. So if you look at the updated cartoon on the left, it's still running through the same template region, but it's now spitting out concatenamers of that 120 base sequence head to tail each time. Now, this was all from Illumina sequencing data, so it's short reads. There was PCR steps involved in preparing the library, so we got a little bit paranoid that those molecular biology steps might create artifacts in the process. So we wanted to look at this problem from a different angle, and we turned to a PCR-free sequencing approach that also allowed us to do long-read sequencing with nanopore. And what I'm showing here is a histogram of the length of these molecules directly from virally infected cells, where we found that, strikingly, these molecules were tens of cDNA repeat units long, where each repeat unit is 120 bases, suggesting that many of these products in the cell are thousands of base pairs long, but still comprise only the same 120 nucleotides being stitched together in this concatenated arrangement. And so this told us that this enzyme is performing a very unprecedented type of reaction that we call rolling circle reverse transcription, where once it gets to the end of the template, there are sequence features that promote template jumping for the reverse transcriptase to jump across back to the beginning of the template so that it can synthesize a second round, spooling out these concatenated head-to-tail cDNA repeats. And then the last clues came about when Stephen looked at the same process in cells that were being infected with the virus. Here, we're now looking at total DNA sequencing. So there's no immunoprecipitation step. In uninfected cells, we don't see that much signal above the background, but a little bit on the bottom strand. But now, strikingly, in the presence of the virus, we not only see DNA being made from the reverse complement of the non-coding RNA, the bottom, but also the complementary strand on the top is being synthesized and amplified in the process. And so this told us that these are now producing double-stranded DNA that has this concatenated arrangement. So now our cartoon is a little more complete. We go from a non-coding RNA to concatenated repeats of cDNA to then a double-stranded molecule in virally infected cells. So that was all very interesting, but it really left us still scratching our heads. What is the point of all this? Why is this good for the cell, and how does this protect against the viral infection? So then Stephen made a second major insight, which came about from him staring at this 120 nucleotide sequence for days or weeks. What is special about this 120 base sequence? And he started to notice some very interesting motifs that gave him some clues about what the function of the sequence might be. The first thing he pointed out to me, and I remember when we met the first time to discuss this, is that one of the three potential reading frames of this 120 nucleotide sequence had no stop codons in it. 120 is also a perfect multiple of 3. And so he kind of started having this idea, could this be getting translated into a functional polypeptide? Because there's no stop codons, and it's exactly a multiple of 3. Now, my first reaction is that could easily come about by chance, because how frequently might you take any 120-mer and find no stop codons in one of the 3 reading frames? So I was pretty skeptical, but then he brought the next observation to my attention, which is that there's a highly conserved motif that precisely matches the -10 promoter box for bacterial promoters. And if you look at the other end of this molecule, a very highly conserved motif that matches the -35 box of a bacterial promoter. Bacteria require -10, -35 boxes for transcription. Now, these promoter boxes are not the right distance apart, but if you now remember that this thing is being synthesized as concatamers, if you look at the junction between 2 repeats, now the -10 and the -35 are the perfect distance apart to complete a promoter across the junction. So the now new hypothesis is that this thing becomes double-stranded and there's a promoter formed between the 2 repeats. So could that lead to transcription? And this was really cool. Stephen already had the RNA sequencing data from the virally infected cells. He just hadn't looked at it the right way. And wouldn't you believe it, but if you looked at the virally infected cells, there was a massive surge of antisense RNA that perfectly overlaid with the cDNA portion of this region. And we also then used northern blotting with a probe that was selective to only hybridize across the junction, and found that 40 minutes after infection, again, a massive increase in the amount of junction spanning RNA signal. And importantly, you can see the length distribution here is from hundreds to many thousands of bases, which perfectly match that long length distribution of the nanopore sequence DNA. So now we're one step closer to this idea that maybe this is a gene that is getting transcribed and maybe translated into protein. So that was the last kind of thing to fall on this project. There's a beautiful genetics experiment in the paper I'm going to skip in the interest of time. But more importantly, even though this provided as close as we thought we needed to get to the proof, we still wanted to directly detect the protein. And so we took our genetic evidence and we went to LC-MS experiment where we were able to directly detect peptides from this newly synthesized gene only in cells with the wild type system, but not a reverse transcriptase mutant, and only in cells that were being infected by the virus, which is called T5. So here's our updated model that the non-coding RNA is reverse transcribed into a first strand cDNA that is concatenated. That is turned into a double-stranded molecule that then creates promoters at all the junctions, leading to transcription and eventually translation into a repetitive protein that we think might look something like this by AlphaFold prediction. And so at some point, we came up with this acronym to start referring to these genes and the proteins as NEO for nearly endless ORF, to describe the fact that these don't have any stop codons in their sequence. So the last big question is, what is immune protective about this protein? And here we had a very clear hypothesis by this point, which came about from our prior knowledge about how this immune system achieves immunity. And this is actually rather common in bacteria, similar to an apoptotic response in a multi-cell eukaryote. With no defense system, a virus will infect one cell, leading to a large burst of hundreds of new viral particles that can then go on to infect all the neighboring cells, causing the population to quickly collapse. There are many defense systems, including DRT2, that actually prevent this by causing cell death or cell growth arrest, where the infected cell is quickly arrested so that none of the viruses can get out and infect the neighbors. The infected cell here, it doesn't survive necessarily, but it arrests its growth quickly enough to prevent the virus from propagating. And so we hypothesized that producing this neoprotein would be necessary and sufficient to drive growth arrest, and that would be the mechanism of immunity for the whole population. So to test that, we got rid of the reverse transcriptase, and we recombinantly cloned neogenes with a normal start and stop codon that were either 1 repeat, 2 repeats, or 3 repeats long. And then we expressed them in bacteria, induced them at a given time point, and asked how the growth of the culture responded. And our hypothesis was that at a certain repeat length with the wild-type neo sequence, we would see growth arresting, just like would happen in an antiviral immune response with the full system. And so here's the resulting data. I'll just focus you first on the most important experimental condition, which was the 3 repeat neo sequence after induction with the wild-type neo, where you see a rapid arrest of growth compared to the empty vector control, or a scrambled sequence that has all the same amino acids, but just in a random order. But then importantly, 2 repeats and 1 repeat of that same exact wild-type neo sequence were not sufficient to cause growth arrest. So this told us that that repetitive nature of the cDNA is critical, because this growth arrest only manifests when the protein product of the gene has at least 3 repeat units of the neo sequence. And again, by arresting growth of the cells, replication of the virus would be prevented, leading to immunity at the population level. So that was where we ended the story. Again, we make this complementary cDNA that is turned into double-stranded DNA, and that is actually a protein-coding gene that's synthesized de novo in the cell, only in virally infected cells, leading to transcription and translation, and that is ultimately what saves the population. There's still a bunch of questions that we don't know the answer to. I think the most obvious one is, why is this specific protein sequence so effective at arresting growth? We don't know. What about the viral infection triggers conversion of this single-stranded cDNA to a gene that can then be transcribed? And then what we're really excited about and focusing a lot of our attention on is expanding from this first version of DRT into the dozens of versions that exist across bacterial species. So just like Cas9 is one of many different Cas nucleases, DRT2 is one sliver of reverse transcriptase immune systems. And we've already found in some of the first projects we're studying that many of these other ones look very different in how they function. And so we think there's going to be a whole universe of new biology to mine from this space. And then I'll just end with a couple of implications that have changed the way we think about the biology a bit. So neogenes are shorter in the way they're encoded in the genome than any algorithm that would automate gene annotation. So it tells us that there could be other genes that kind of don't conform with the expectation we would have for what a gene should look like. These genes are not encoded in the chromosome, in a plasmid, in a viral genome. They're only made in this transient state that doesn't exist in its full-length form. So you can't look for the gene in the genome the way that we typically would expect to. These genes don't get encoded in a linear version the way that all bacterial and eukaryotic genes do, whether they're spliced or unspliced. These are circularly permuted, which makes their discovery also quite challenging. It complicates the notion of how genes are typically expressed. You really need to go back and forth between multiple stages of DNA and RNA before the neo gene reaches its final intact state. And then again, we're excited to start thinking about how we might be able to exploit this enzymatic ability to amplify a sequence in many copies from a very controllable non-coding RNA starting point to synthesize DNA in the context of potentially some other tools in the space that combine targeting strategies like CRISPR with reverse transcriptases. And then finally, we've really started thinking a lot more about whether there might be other similarly cryptic genes that are elsewhere hidden in the genome, perhaps even outside of bacteria, and how we might use some of the approaches we developed here to go looking for them. So last slide, just coming back to why we think transposons are a really exciting place to look for new biology. They're far more than simple selfish parasites. So much biology has flowed out of the genes that transposons encode. This gene exaptation is a very common theme in biology, and it's been the source of a lot of interesting and exciting RNA-guided and RNA-templated systems, and we think that there's going to be many more of them that await discovery. So with that, I'll end by thanking a really fantastic group of scientists in the lab, and thanks very much for this kind invitation. I'm really excited to take some questions and meet many of you later today. Thank you.
Thank you, Sam. This was wonderful. And as we go and people collect their thoughts, why this system was lost in evolution? Both what you described today.
Well, I guess we would hope that maybe it wasn't lost, and we might be able to find similar examples of domesticated reverse transcriptases in other eukaryotes. You know, we recently started annotating all the reverse transcriptase genes in the human genome. There's tens of thousands, which is not surprising because there's millions of copies of ALU elements, there's hundreds of thousands of copies of line elements. And I think most people would look at lines and say the ones that are active are those that are actively jumping. But might there be reverse transcriptases that get expressed that have functions in the cell that does not involve retrotransposition, but similarly creating some gene in the cell? So that's the kind of idea that we're wondering, is it worth following up on? And it might say perhaps that kind of function hasn't been lost and it's just not something we've looked for or found yet. So that's what I hope for. We'll find out.
Thank you.
Thank you for that talk. My question had to go with the first part of your talk, and I was wondering if we know what determines the arrays in your Cas systems.
Yeah, I love that question. So as you might know, it sounds like you're quite knowledgeable in the field. The Cas-transposons have lost the machinery to integrate new spacers. So most CRISPR systems encode enzymes like Cas1 that integrate new spacers and grow the CRISPR array over time. Cas-transposons do not have those integrases. But we've done comparative genomics to show that between very closely related transposons, there is diversity in the CRISPR arrays. So our best guess is that they will occasionally rely on similar CRISPR systems elsewhere in the genome that do have those machinery to kind of act in trans to update the spacer information in the CRISPR array. What's still quite murky is like, what are the guides? Because most of them don't match anything that's in the NCBI reference genome. The few that do usually are found in plasmids. And so our current best guess is that these CRISPR arrays are kind of packed with guides targeting mobile conjugative plasmids so that the transposon has a way from getting from the genome onto a mobile piece of DNA that can be spread between bacteria. It basically gives it another life in a different species if it can get out of the cell. And so we think that the guides are probably often targeting mobile plasmids, but no one has actually studied that in the lab yet. So I think it's an exciting area for the future. Thank you.
Thank you very much for presenting this wonderful work. My question is with the Cas system, how specific are those? Are they much better than regular Cas9 or not having off-target or anything?
Yeah, very good question. So the wild type Cas systems, I think, are exquisitely accurate. Not all of them, some of them. The ones that we have chosen to exploit for human cell applications show very excellent specificity parameters in bacteria. We do see a slight decrease in specificity as a function of directed evolution because we're not selecting against a loss of specificity. And so now we're doing some reversion analysis where we see which mutations can we maybe pull out to protect and maintain the high on-target specificity. It depends on how you count it, but we've compared the accuracy in terms of counting on and off-target events to PASSAGE, which is a prime editing and recombinase-based method, and Cas9 nuclease, and they're similar, but we do see an increase in non-RNA guided DNA integration with the evolved variants. And so that's something that is going to be an important future engineering goal.
Yeah, it's really fascinating talk. So continue on the cast. Is the limitation of the cargo size? I mean, do you have an idea about how big DNA you can insert?
Yeah, so we've gone up to 10 kb and see no decrease. With 15 kb, we saw a slight decrease, but importantly there, we were delivering the donor as a mass normalized amount per experiment. And obviously, as the DNA payload gets bigger, the number of molecules of the plasmid will go down. So we didn't control for like the stoichiometry or the concentration of DNA. So we think it's not going to be very sensitive to size, and we've seen the same thing in bacteria. So I would like to say within a reasonable limit of what is easily deliverable, there's probably not going to be a major dependence on size. And we show that at least across a window of hundreds to 10,000 base pairs.
Hi, really good work. Thank you. My question is about the NEO system. So it seems to be kind of an inefficient thing. You go from a DNA to RNA to DNA to RNA before protein. So what's the advantage of this over the traditional just, DNA, RNA, protein?
Yeah, I'm curious how Ben would answer this question. I guess I would say like evolution doesn't always take very logical or kind of sensible paths, nor does it need to. It just does what works. It'd be really cool if you could follow what the intermediates were leading up to this immune system, because something about those intermediates needed to be useful enough for it to continue being selected in the way that it was. But I mean, I would say there's a number of now very exciting and interesting immune pathways that don't look very logical, but they work. So that's like my kind of BS answer to your question. I will say maybe one thing which you didn't ask directly, but another question is why go through this whole crazy process of concatenated DNA synthesis? We think that the NEO genes are probably so toxic that encoding them in their full-length form, like you can imagine it'd be much easier to just have the NEO gene controlled by a really sensitive promoter that is only turned on in phage-infected cells. But in bacteria, most things get expressed at some low leaky level. And we think that the neotoxicity is probably severe enough that it's too dangerous for the cell to encode that gene in its intact state, because you'd risk autoimmunity where like low level of expression would cause growth arrest and the cell dies, even though it hasn't been triggered by a viral infection. And so cells need to always balance immune system diversity with a low level risk of autoimmunity. And we would hypothesize that maybe this convoluted process of creating this gene only in virally infected cells was nature's solution to that problem.
Wonderful. Mic was off, sorry.
Yep.
Hello, wonderful talk. I had more of a theoretical question. I was curious, so your Cas systems lost the ability to be reprogrammed. Do you have any sort of speculation on what the ecological niche of a very specific but not necessarily programmable transposase is? I mean, line 1 is incredibly successful, right? Why did this evolve or potentially go extinct?
I love that question. I guess on the retrotransposon front, I would answer with R2. Do you know about R2 retrotransposons? Those are in the same family of doing targeted prime reverse transcription, also non-LTR, and those are highly specific for ribosomal RNA loci. So they are successful at integrating only in one place in the genome, which is rDNA sites. Now, rDNAs are present in many, many copies in eukaryotic genomes. But I guess, I think different mobile elements will have found different strategies for survival. In a different version of my talk, I have kind of like a spectrum from highly specific to completely random. And I think multiple different points along that spectrum can be evolutionarily successful. Viruses, for example, often use integrases that integrate at only one attachment site. So the downside is if the host you infect doesn't have the attachment site, you have no way to infect in a dormant fashion. But the benefit is if you recognize a site that is extremely conserved, maybe a site that the cell can't mutate because it's important for some other central process. Like it's a catch-22 situation. And we think that these transposons are probably using similar strategies to target sites that are probably fixed in some way because they're highly conserved in the cell or in the plasmids that they target. But yeah, I think across even the TN7 family, there are some that are RNA guided and don't have a lot of different guides. Then there are some that don't have CRISPR at all, and they're quite random. And so within just that one superfamily, we see many different solutions to this question of specificity and promiscuity.
Hi, very nice talk.
There's something special about this side of the room here. Maybe it's only
we're just starting here. Yeah, my question is about the mechanism of DRT2 and its ability to perform rolling reverse transcription. How is it that it's able to jump from the 5' to the 3'? And is it necessary that those need to be close in distance? Have you disrupted that stem loop to see if the rolling reverse transcription also occurs? I was also curious about the interactors of neoproteins. Is it that it's expressed in high levels and it's creating some sort of toxicity and that's why it's allowing for growth arrest, or is it that is it interacting with some other proteins to allow for growth arrest?
Yeah, those are wonderful questions. Like, come to our group meeting, please, because those are the things we talk about every week. On the second question, we unfortunately, we actually don't have answers to those questions yet, and I'm not hiding some unpublished data. I like the idea that there's a host protein that it might be sponging up or sequestering. Like you have a highly specific binding interface, maybe at the junction between two repeats that now sucks up transcription factor or some critical translation factor. My argument possibly against that hypothesis is that the neo that we tested in E. coli comes from Klebsiella pneumoniae, and we found that the toxicity is quite broad host range. So either those potential protein partners are highly conserved, or there aren't host factors that it recognizes, because otherwise you might expect it to be more selective for only toxicity in the host. We also, we have tried, we are now fishing it out of cells to see if it pulls down any other protein partners. That was a tough experiment to even set up because we have a very hard time detecting it. Mass spec, very sensitive. If we put an epitope tag on it, we break the immune system. If we put an epitope tag on the recombinant gene, we can basically barely detect it, which tells us that it seems to be driving arrest very soon after its initial production. So it's not accumulating at large levels. I have one pet theory that maybe it's like arresting ribosomes, because then you imagine as soon as you get, you exit the nascent polypeptide channel, maybe you sit on the ribosome and you plug up that channel from being recycled. And that would be a very quick acting way to basically pull all the active ribosomes out of the population. But we have no evidence for that. On the first question, we identified in the paper this critical trinucleotide motif ACA right here and right here. And we showed evidence that I think more or less proves the idea that there's a likely switch in hybridization between the cDNA that first is templated by these three ACA nucleotides and then probably reanneals to these three, so that now you've basically swapped where that annealing guides the reverse transcriptase to incorporate the next nucleotide, and that would get you from the end back to the beginning. We have now made some more mutations that suggest that as long as you maintain nucleotide identity between these three and those three, that's all you need for template jumping. But I think we need to test a larger panel to really be certain of it. I would love to also do more biochemical or biophysical experiments to look at template jumping, because I think there's probably going to be very interesting kind of enzymatic property there that permits that. And I'm also really curious if the reverse transcriptase has something special that promotes this, or is it all about the non-coding RNA? Could you take another reverse transcriptase, give it this structured RNA, and the RNA intrinsically drives template jumping, or is the reverse transcriptase somehow adapted to be really good at this? Because many other reverse transcriptases, like what we use for RNA sequencing library prep, if you give them RNA, like they will do non-templated addition. Sometimes there are certain reverse transcriptases that are commercialized in kits to do template jumping as a different way of getting adapters ligated onto your cDNAs. So maybe this property is shared with other reverse transcriptases too.
Hi, Dr. Sternberg. I'm Rafał. Nice to meet you. Wonderful talk. I have a question. This cast integration was demonstrated in vivo, or what are the hurdles that prevent from this?
Yes, this was all. So I always joke when I worked 1 year at a biotech company, my entire training in vivo was like in a cell. I know like in this audience, that's not what in vivo means, but it took me more training to like figure that out. And at Caribou, where I worked for a year, we were very careful to not use either in vitro or in vivo for a biochemical experiment because it's just inherently confusing. So yeah, everything we showed was in cell culture. We've now shown activity in HEK cells, HeLa cells, K562s. We've done recent experiments during revisions in primary fibroblasts, but nothing in vivo. And I think the biggest hurdle there is going to be delivery. Also, everything I showed was with very kind of brute force plasmid co-transfection. So I think the real next challenge, once we feel like we've hit the best Cas system to move to the next stage, will be how do we deliver this in a way that's going to be less cytotoxic. The DNA needs to be DNA, but we could probably deliver the other components as either mRNA or perhaps RNP complexes. So that's something that will be kind of like in the future research for us.
Great talk. I have, I guess, 2 questions about NEO. One is kind of from a population evolution standpoint. In a population, how many of the bacteria will be expressing NEO? Because you can imagine that it imposes like a fitness cost to kind of produce these long cDNAs and RNAs. And if you have cheaters in your population, that could you could have an advantage over your fellow population.
Cheaters that don't make NEO somehow. So I think, yeah, you ask a good question, and I'll acknowledge here that like many in the phage defense field, we're working in highly non-physiological conditions, which is one monoculture with like a usually unphysiological MOI, because in nature, viruses are probably blooming like crazy. They'll be extremely rare, and then they find some niche, and then all of a sudden you have a very localized, massive bloom of virus. And here we're like monoculture, there's no biofilm, there's no microbial community. And then we just dump on like a clonal phage population. So I guess I wouldn't probably want to extrapolate too much from how we did things to directly answer anything about your question from our experiments. I think the cheater scenario, like, I guess I would imagine that if the phage is successfully infecting a population, those cheaters that don't make the long toxic neogene are going to get killed off by the phage. And so that's the pressure to maintain and evolve the system in the first place, is that the real threat is elimination by the viral population if you don't have the right synergy of immune systems. But, one thing that Rodham Sork in the phage defense field would argue is that immune system repertoire is probably highly dynamic. Bacteria gain and lose systems quite rapidly, often through horizontal gene transfer. So it's probably the case that the low-level toxicity would be mitigated in cells that are not actively being infected, because you'll now occasionally just lose the immune system through a horizontal gene transfer event or a recombination event. And so probably you do get cheaters that just lose the system if they're not actively being selected by phage.
Hi, Dr. Sternberg. Thank you so much for interesting talk. So my question is about the Cas systems. So you showed these impressive and specific integration events, but I kept remembering of these old CAR-T engineering papers about this SB transposase and about this theoretical risk of insertional mutagenesis. when you have these specific iteration events, how much of the DNA sequence fidelity is maintained because you have such a high, bigger cargo size that is being integrated instead of maybe installing some putative mutagenesis sites?
Yeah, no, it's a great question. And I think I took the slide out, but we've done genome-wide sequencing in mammalian cells. It wasn't quite as easy as bacteria, but we do see, as I mentioned earlier, with the evolved Cas system, we do see an increased incidence of low-level random insertions. And those are, we think, CRISPR independent because we get a similar low-level integration spectrum if we just remove the CRISPR components and just express the evolved transposase components. So we think that we're probably selecting for a slight loss of fidelity in the accuracy. And again, that's something that we're now hoping we can take what the directed evolution identified and now use like more manual engineering to figure out which parts to keep for the efficiency while like restoring a little bit more of the on-target accuracy. We have also, another thing we would want to be looking at carefully in the future is, plasmids will also randomly integrate at sites of double-strand breaks. So I think, in the end, different ways of delivering DNA that maybe don't have a full circular plasmid, we have kind of chemically modified linear DNAs that have a lower incidence of random integration like that would be something also that we want to explore in the future.
Sam, I know we could go forever, but I will limit for a question more.
Great. I love the questions, by the way. This is such an active and engaged audience. It's quite remarkable, actually.
Hi, Dr. Sternberg. Thank you for your talk, and I'm Olivia from Audrey's lab. And my question is about your first part of the talk regarding the transposon. And so it's just so impressive that the transposon, the CRISPR-associated transposon does not depend on any cellular repair pathways. But my question is, like, as you may know that for most of those mammalian cells, the cellular repair pathways are way more sophisticated and they are much more sensitive and cautious for any kinds of like nicks or breaks on genome. So in your scenario, have you ever investigated any cases that there will be some competition between any of those cellular pathways with a efficient DNA integration?
Yeah, really good question. So one thing we looked at, like a simple and immediate thing we looked at is whether we get increased indels at the sites we target, because, I also don't think I have like a more specific picture of the Cas transposition pathway, but there is some reliance on DNA repair, not double-strand break, HDR repair, but similar to prime editing, we end up with single-strand gaps at both ends of the integrated transposon that needs to be filled in and then ligated back together. So there will be a reliance on some of the same kind of enzymes that you would need to like resolve the flaps in a prime editing intermediate. We look first if we're getting indel formation at the sites of integration and we see no increase above like background sequencing error. That is in contrast. We've now done head-to-head comparisons of cast and passage. Passage is similar to paste, which is the combination of prime editing with a large serine recombinase to do landing pad installation. And so there, prime editing will generate some indels if you don't have complete prime editing product formation. And so if we compare cast to those other methods, we see basically undetectable indels with cast, and we can still see reasonable indel formation from prime editing methods. So we think we're better off there, but we're still at an efficiency range where I don't want to say there aren't any other outcomes, because I think until we're 50 to 80%, it could be that it's rare and it's below our detection limit. But I think it looks promising, and based on what we know chemically about how the transposition reaction works, there shouldn't be a major risk of kind of unwanted byproducts the way that there is with most of the other editing pathways. Thank you.
Thank you so much for that really interesting talk. So I was kind of curious if you've tested the Cas system in known regions of varying like chromatin accessibility or DNA methylation, and whether that influences the transposase activity.
Yeah, go ahead.
I was kind of thinking like, oh, this might be an argument for kind of developing a library of target sites that can cut around difficult areas to even, do site-specific editing.
Yeah,
like that.
I was going to answer your first question with your second
sorry.
comment, which is like, we would love to now do library-based experiments to answer your first question more systematically. Because now, because it's very programmable, we can just say, well, instead of targeting a couple sites that are euchromatin or heterochromatin, let's take 10,000 guides or 50,000 guides, and we're going to just hit all the sites that are like at varying levels of chromatin accessibility, and then just empirically determine what's the relationship there. So we haven't done that yet, and we've, as you could probably tell from the sites we've hit, those are all like known to be open. They're the sites that everyone in the gene editing field picks first, HEC3, AVS1, et cetera. So we went for the low-hanging fruit, easy and well-characterized sites first, but I think that would be like one of our next things to go after.
I have two questions. One about NEO-system. So you show that you have AlphaFold predictions of these proteins. Did you try to do structure search, and is there any similar enzymes that have similar domains or proteins that have similar domains?
Yeah, we did, and we're increasingly big fans of fold-seq and of like structure-based homology searching. Nothing comes up from that aside from I think Dolly search reveals similarity to heat repeat proteins, which are similar alpha helical, like solenoid proteins. I don't think there's likely to be a common ancestor there. So yeah, unfortunately, that approach didn't reveal some like smoking gun clue for what's going on and what the origin of this protein is. I would guess it's probably like a de novo gene that doesn't have ancestral relatedness to other genes. But also there's not, I mean, AlphaFold is pretty reliant on like multiple sequence alignments, which don't exist for this because no one else has annotated these genes before. So I think also maybe with more information in the future, we'll have a better starting point for that analysis.
Second question is, I just want to maybe hear your opinion or take on insertion of large sequences in a way as a therapeutic approach to, let's say, address some of the diseases that are caused by hundreds of mutations, right? So the idea right now is to use these technologies to insert these large sequences into some type of safe harbors. Do you think that's a viable strategy for therapeutics, especially considering that now you're inserting a gene that has totally different regulatory sequences around? Or do you think we should think about, in a way, replacing the broken gene?
Yeah. There's probably many people in the audience that are far more qualified to answer that question than me. I think we would probably promote the idea of inserting the cDNA at the endogenous locus, the way that I showed in one of the cartoons, where you would still capture the endogenous regulation of the gene, and you'd include like polyadenylation sites so that the downstream gene is basically not transcribed or expressed anymore. But of course, what we'd rather do is replace. So if we had a magical recombinase that could swap out the entire sequence, like that would be more desirable than inserting in front of it and silencing everything past it. Some of you will have seen the work on IS-10, and by the way, we referenced your NOT5 structure. We'll talk about that later. I think that's a really interesting system. We'll have to see like how well that also ports over to mammalian cells. That has some potential opportunities for replacing, but I think has its own like list of idiosyncrasies to work around. I know Fyodor Ornov is pushing a lot this idea of safe harbor insertion for like any disease, and I think the downside is, as you mentioned, you don't have the endogenous regulation, and it may not be sufficient to have one fixed expression level in one fixed locus for different kinds of pathologies. So we like the idea of going into the native locus, and with this kind of a system, it's not going to be difficult to redesign the guide to do that.
Last one.
Hi, I have a general question about retrotransposons. There is one model of retinal degeneration, which is caused, like a mouse model of retinal degeneration, which is caused by migration of retrotransposon called L1 to disrupt one of the genes responsible for development of photoreceptors. So we have been keeping this mouse model, for few years, let's say. And then with time, we started to notice that some of the mice, the phenotypical characteristics of this disease model kind of started to disappear. Is it a reasonable possibility that that copy of retro-transposon that was disrupting this gene got migrated to another place without leaving any traces? Or is it very unlikely, within the few mouse generations?
Classical retrotransposons don't have a way to excise themselves at the DNA level. They can they go through copy and paste and they don't excise during the course of transposition the way that a cut and paste DNA transposon would. So not to my knowledge. I wonder, is it a heterozygous insertion? Because could there be low-level recombination with the sister chromosome to basically reinstall the vacant site? That'd be my very naive question back at you. But I don't know of any known retrotransposon pathway for elimination at the DNA level once it's retrotransposed somewhere. But it sounds interesting.
Sam, thank you so much.
Thank you very much.
I hope you enjoyed the visit, and thank you for your wonderful talk.